I'm going to talk about some of the challenges that we have in TB drug development and some of the answers that we give to the questions that we are faced with. And I would like to make some suggestions about the way that we discuss those questions and how we come to perhaps some conclusions. And involved in that will be, I'll give you a quick overview of the PREDICT-TB consortium for those of you who are not familiar with our work. Then I'll talk about the review of clinical trials that we've carried out in order to facilitate our understanding of preclinical development. Our general approach to preclinical experiments, which is a little bit different perhaps from what some people are used to. And then finally, talk about the role of evidence synthesis, both in preclinical experiments and how we can evaluate the usefulness of preclinical approaches using meta-analytic methods. So to start at the beginning, I think I probably don't need to emphasize for this audience many of the challenges that we face in transitioning from preclinical development, particularly into early clinical development. So not only are there significant disconnects between in vitro and in vivo work involving all of the problems that are listed there, but in particular, in the transition from in vivo work into phase 2, there are many uncertainties, and a number of those we've touched upon this morning. But in particular, our understanding of the PK and the dosing and PK-PD relationships and the pharmacodynamic model that we use in the in vivo phase, is that really applicable to the kind of data that we can generate in phase 2, and how can we understand really the connection between the two? So PREDICT-TB is a response to that problem. And the way that PREDICT-TB is structured, it's structured around a set of 3 overarching goals, really. So it's a very large consortium involving 21 partners across Europe, 3 pharmaceutical companies, and a large number of academic institutions. And the overarching goal is to evaluate a representative set of anti-tuberculosis drugs, all of the drugs that you would think of in first-line therapy, and many that are now being proposed for use in first-line therapy, and to evaluate that in the widest possible set of preclinical model systems. But not only that, we'd like to do that with reference to existing data from clinical trials and to compare the two directly with each other. Ultimately, what we'd like to do on the basis of this exercise would be to define the currently optimal critical path, to make some changes to that in preclinical development. And very central to our approach, and you've seen many examples of that this morning, has been the use of a mathematical modelling approach to the data and to try to integrate the data and to create a framework that will enable us to do drug development better. And this is a partnership amongst many of the people sitting in this room, involving a number of different work packages. And the one thing I'd like to particularly draw attention to is that it's a multidisciplinary consortium. So this is one of the few fora, I think, where people can sit down, people who work in test tubes, people who work in animals, people who do clinical trials, people who do PK-PD analysis. All of those people are in this consortium and talking to each other in a structured and productive way. So that's roughly speaking how the consortium is constituted. And this has been running now for about 4 years, and in the first 4 years of the project we've evaluated a set of 14 drugs, starting with the existing first-line agents and evaluating the performance of those agents in preclinical systems, modelling that data, having a look at the results, deciding whether the data is what we need to actually be able to extract the information from those systems that would be useful in drug development. There were some refinements made both to the assays themselves and also to the sampling schemes in those assays and the models that we use for them. And then in round 2, we've begun to look at, novel agents, what were considered novel agents 5 years ago. So rifapentine, moxifloxacin, many of the drugs that you heard about this morning, and also some of the newer agents such as bedaquiline, PA824, and so forth. And at the end of this process, in about a year's time, we hope to have a comprehensive description of the performance of all those assays across that set of drugs, and to be able to say something about the relative performance of all of those systems across that whole set of data. So to take the first step first, one of the things that we were struck by at the outset of the consortium was, well, if you want to know how well preclinical systems work, what's your target? How are you going to know when they've worked? And so one of the things that we set out to do was to try and understand what the literature shows in terms of tuberculosis clinical trials about the performance of all of these regimens. Now, I understand that after lunch, especially if your eyesight is not very good, this is bound to put you to sleep. But it's to illustrate the point that there is quite a lot of data out there, and data for which, before we did this, there wasn't a great synthesis. There wasn't a go-to reference that you could look at and say, okay, this is how this drug performs in clinical trials. So here you can see some of the outputs from that work, which is shortly to be published. On the left there, you can see pretty much all the existing published data in the literature that relates to one of the standard measures that we use in very early clinical development in phase 2A, the so-called study of early bactericidal activity, which was originally performed just over the first 2 days of therapy. And because that's been very, very widely reported, we were able to extract a lot of that data from the manuscripts, and you can see that presented here, and ranked in order of efficacy. So you can see that, I don't know whether you can see actually, but you can probably see that down at the bottom there, one of the first EBA studies that was first ever conducted with rifabutin is paradoxically one of the drugs that has almost no EBA. And if you rank those regimens in order of their efficacy, you come down to the ones that perhaps you would be expecting to see: HRZE, SHRZ, isoniazid-based regimens, and so on. And that data covers about 32 different combinations of drugs, single drugs and combinations of drugs. And on the right there, you can see the data from the more traditional, more recently accepted measure as a surrogate endpoint, 8-week culture conversion. And that's data taken from phase 2 trials and from phase 3 trials that also report intermediate bacteriological endpoints. And again, quite a large data set, more than 20,000 patients in the literature, more than 45 combinations. So there's quite a lot of data out there that enables us to try to rank regimens and to understand how they actually perform in the clinic. And that's the target that we're trying to hit with our preclinical systems. When we compare our preclinical systems with these results, that's what will tell us whether or not they're giving us information that's useful in drug development. So that all sounds fine and dandy, but in designing this project, we had to be somewhat pragmatic. So if you look at all of this data, you can see that there are huge numbers of combinations, huge numbers of patients, quite a large number of studies. And again, you won't be able to read all those different drug combinations at the bottom. But we selected from those combinations the most commonly studied regimens and what we thought were the most important, that pragmatically everybody would have time to study in their system. And in fact, during the first round of therapy, about 30% of the majority of the partners in the consortium covered at least a third of that space. So this was a very, very significant effort in terms of trying to evaluate the biggest number of drug combinations that you possibly could in your preclinical system. But just to make the point again, we calibrated this very, very carefully to the data that we knew was out there in the clinical literature so that we would be able to know or be able to judge the significance of those results. So I'm going to take one example just to illustrate this approach and what can and can't be done with it. And I'm going to share with you some fairly hot data. Perhaps I hope it's not so hot that I'm going to get burned by presenting this, but I'm going to try. And I'll value your feedback and questions. So I said that we took a slightly different approach to preclinical data. And I think you've seen a lot of this on the posters. What we said to the experimentalists was, we'd like you to do your experiments a little bit differently. And you can imagine how that was likely to go down with people who'd been doing these kind of experiments for years. But our experience actually was very interesting. People were very open to the concept of changes in experimental design, to the concept that the kind of analysis that would be done on their data was not a kind of analysis that they would be capable of doing themselves. But they were trusting of that, and they worked very successfully with our Work Package 5 and the statistical modelers in Work Package 5 to do these experiments differently, and more importantly, to derive from those experiments endpoints that would be comparable with clinical trials. Because one of the problems in the literature and the systematic review is that everybody tends to report slightly different things in the historical literature. So our aim was to look at the data from each of these experiments and to do some statistical modeling on them so that they would be, broadly speaking, comparable from experiment to experiment. And here's an example of that. This is a very simple example, but this is just some quantitative bacteriological data from a mouse experiment. And you can see on the far left there after the inoculum has been delivered, the bacteria is starting to grow, chemotherapy is delivered at the peak of that curve, and then there's a steady decline in colony-forming units over the successive weeks. And oftentimes that data would be described as what dose achieved a 2-log drop, or perhaps what was the proportion of mice that were culture negative at a certain time point. But our approach was to compute the elimination rate of organisms. So we fitted some very, very simple mathematical models to this data in order to derive elimination rates and to be able to rank the regimens on that basis. And you can see an example of that on the right there. And this is quite complex data as well, so there are often discussions about whether you should measure the lungs or the spleen, but we found that, broadly speaking, the elimination rates and the ranks based on those elimination rates were rather similar. So much of what you're about to see is based on this idea, okay? So we're taking all of these experiments and trying to analyze them in a way that's comparable. So in doing that, it enables us to look across that whole space of combinations that I showed you, and to look at all the mouse experiments that have been performed, both in the literature but within the consortium. And we're also very fortunate to have some data brought into the consortium by external partners, which has been a great step forward, I think. And you can produce plots a little bit like this, which again, you have to have good eyes to see all the detail. But essentially, this is a very simple meta-analysis of all the mouse experiments that have been done within the consortium and all the data that we have, to which we could apply that modelling approach, and pooling that data to give an overall estimate of how well that regimen generally performs in the mouse. Now, there is a caveat to that, of course. A single estimate may or may not be appropriate given the different characteristics of different mouse models, and you can see that for several of these regimens, the heterogeneity is relatively high. So we're having to use a random effects model to reflect that in this simple kind of mouse meta-analysis. But it enables us to see how that regimen performs over 4 or 5, 6, 7, 8 different replications of that same experiment in different mouse models. But that's not where it stops. So what we can also do is do what I showed you with the phase 2 regimens. We can rank these regimens one against the other on a pooled estimate and see whether the overall rank order in the mouse models agrees or not with the phase 2 data. Now, there are some problems with this because there are some missing regimens. Some of the regimens that are covered in the clinical data set are not covered by the mice, and sometimes vice versa. But broadly speaking, you can see, and this is data on more than 1,500 mice, but that it covers more or less a space of combinations that's rather similar to what I showed you with the phase 2 data. So we're talking about 32 different combinations of drugs here, which should be enough for us to be able to understand the variability in drug response. So once we can do that, the next question is, well, okay, that's fine, but what happens when you compare the one with the other? And that's what we're really wanting to do. And so this is an approach that's been developed by the Camarades collaboration, who are responsible for trying to improve the quality of preclinical animal experiments. And the typical approach is simply to compare the endpoints that you get in a clinical trial with the endpoints that you see in an animal model. And this is what you can see here. So in red, we're showing the estimates that are derived from modeling the clinical data. So these are not only estimates derived from the manuscript, these are individual patient data that we've been able to obtain and to model in essentially the same way that we model the mouse data. And this plot, actually, I say that, that's what we're doing next. This plot is actually based on what's reported in the papers. So if you look here, the endpoint that's reported for the clinical trials is the classical EBA 0 to 2. But the numbers are not what you're used to because they're rescaled to make them comparable with what you see in mice, which is numbers of CFU per mil per week. Okay, and I can see a number of open mouths around the room because what you see is that the rate of elimination in the mice is much, much less than you see in an EBA 0 to 2 study. Some people may be surprised by that. Some people may not be surprised. So those people who say, well, doing a mouse experiment is just like doing a kind of furry EBA study, I don't think that's right, actually. It's interesting that EBA 0 to 2 elimination rates are much, much higher than the elimination rates that you see in the mouse. Perhaps because, as has often been said, EBA studies probably study the effect on extracellular bacilli, whereas many mouse models, of course, don't study that. They probably study the net effect on mostly intracellular bacilli. So there's a bit of a disconnect there between one of the traditional measures that we typically use in early drug development and what animal models are telling us. If you look in the literature, one of the problems that I referred to in mice is equally prevalent in clinical data. So particularly in phase 2 studies, we reported in trials last year that the quality of reporting in phase 2 studies is so variable and so different from study to study that it's almost impossible to combine the data. And so in fact, the number of regimens that we can clearly compare on some of the more prolonged measures of early bactericidal activity is relatively small. But this is one of them. So this is a subgroup analysis just looking at the first-line regimen HRZE. And what you can see is that when you prolong the analysis over the first 14 days, so a 2-week EBA study rather than a 2-day EBA study, you begin to see somewhat closer rates of elimination. And though we only have 3 mouse studies pooled in this particular instance, you can see that they're not the same, but they're more or less in the same general vicinity, which is very different from the picture that you see with EBA 0 to 2. But the picture becomes even clearer, I think, when you start to think about actually modelling the clinical data. So this is not taking results of clinical trials from manuscripts. This is actually using the individual patient data and applying the same modelling approach to the clinical trial as you do to the animal model. And when you pull those estimates, because it accounts for some of the flexibility and the changing patterns of drug response, you actually see much closer agreement between the results of the mouse models and the results of the clinical trials. So right at the top there, you can see one of the very traditional regimens, this is the SHRZ regimen, and you can see that the rate of elimination in clinical trials is of the order of, in one study, certainly about minus 0.8 per week, whereas the mouse studies are more or less the same. And if you look down the plots, you can see that regimen to regimen there is some variability, and that not all of them agree very closely, but that some actually are in fairly close agreement. And this is the approach that we're taking forward and will be completed in the coming few months, that will enable us to look directly at the results of clinical trials and the results of mouse experiments in this case. And just to conclude, we can also look at this in a slightly different way. And here's another way of looking at that. And this is looking, in fact, now at 2-week culture conversion and just trying to understand the heterogeneity that you see from mouse model to mouse model. Because I said that the I-squared values in the meta-analysis were quite high. So it seems very important to understand what it is about those individual mouse models that makes them vary so much. We are not making a judgment about whether one model is correct or another, but we'd like to know what their characteristics are so that we can try to understand how to reduce that variability, perhaps. And what you can see here is a very simple plot, a dot plot really, of the results of 8-week culture conversion on the right-hand side, and at the bottom you can see the elimination rates calculated from mice. And the overall trend is quite interesting because though there is a lot of variability, you can see that some of the really terrible regimens that were used in the 1950s, like PAS, actually have almost no activity in mice at all. But then when you come up to the top of the plot, where you're looking at modern short-course chemotherapy regimens, SHRZ, HRZE, the mean estimate is much greater than that. And so, broadly speaking, just using the eyeball test, there does seem to be some measure of agreement between those measurements on the basis of the clinical potency of the regimen. And finally, we can look at this in a proper bivariate format, so we can start to construct confidence intervals around those pooled estimates for each regimen, taking into account all of the heterogeneity and the variability that I've shown you, and do a meta-regression analysis that enables us to look at the relationship between mouse elimination rates and culture conversion in humans. And this is an early draft of one of those plots. And again, you can see, broadly speaking, that there is a relationship between those two things, but the variability is very, very high, and the confidence intervals are very, very wide. But as we add data to this analysis, more data to this analysis, it will become more powerful, and it will be easier for us to see whether those relationships are statistically reliable or not. So, in my view, the arguments that we have, and we have them all the time, in meetings like this about the value or lack of value of animal models and any other preclinical system that you care to mention need to be based on a full understanding of the evidence. And what we're trying to show here is that an individual level data approach really facilitates what I would call the commensurability and the meaningfulness of those comparisons with clinical trials, because we're trying to compare the same endpoints, we're trying to scale them so that they can be looked at directly together. On the whole, and though this is early data, the mouse elimination rates, I think, do perform better when we use similar analytical approaches and time periods to those that are used in a statistical modelling approach to clinical trials, rather than some of the things that are reported in the literature which seem less similar. And we plan to extend this approach to other preclinical systems and indeed in the future to relapse experiments to try and fully understand the panoply of preclinical systems and how they stack up with the results of clinical trials. And this is one aspect, I say it's not the only aspect, but it's one aspect of the work of PredictTB that I hope in the future will make people look back and say, you know, perhaps this is the beginning of a more rational basis for understanding what we do in preclinical development and also for planning future research. So I'd just like to end by thanking everybody who's been involved in this, because it's a huge effort and it's not my work. This is everybody's work in the consortium. But I'm going to single out a few people in my group who've helped with the modelling, particularly Henry, Alessandro and Laura Bonnet, who is a statistician. And I'd also like to thank the experimentalists who worked so hard to generate all of this data. So particularly Jurian, Janmin, Walter van den Broek at Janssen, Stefan Kaufmann at Max Planck. And finally also to Anne and Eric, who've engaged very positively with this effort. And we've begun to see that collaboration, I think, can yield results. So thank you very much. I think I still have a little bit of time for questions, maybe.
Yes.